All right, it looks like everybody has managed to find their way into us, so let's get started. I'd like to welcome everyone to the Vasculitis Foundation webinar today. I'm Kathy Olevsky, the host for the Vasculitis Foundation's educational webinar series, but I'm also a patient living with MPA vasculitis. These webinars are part of the Vasculitis Foundation's commitment to patient education. And today's webinar is about the microbiome and vasculitis with Dr. Rennie Ree. After we finish this webinar today, there'll be a recorded version on the Vasculitis Foundation's YouTube channel, and everyone who's registered today will receive an email with a link to it just as soon as it becomes available. Before we get started today, I'd like to just go over a few details. Because this is a live webinar, we have muted everyone except our guest speaker. In today's webinar, Dr. Ree will do a presentation, and after her presentation, you all have the opportunity to ask her questions. These questions can be asked by typing your questions into the Q&A box. So if you don't see the Q&A box right now, click the three dots on the far right of your screen in the bottom where it says More. Please make sure your question is a general question about the microbiome and vasculitis, as Dr. Ree cannot comment on your specific medical situation. Also, if you need closed captioning, you should look for the box at the bottom. Usually it's on the bottom left corner, but if you don't see it there, also look under the word More, and you might find it there. And one more thing, there's a chat box. So the chat box is to talk with other people that are watching the webinar or the Vasculitis Foundation staff in the background. But remember, use the Q&A box for your questions. And you'll have plenty of time to think of those questions during the presentation. And now that all that housekeeping is done, let me introduce our guest today. Dr. Rennie Ree is a rheumatologist at the University of Pennsylvania and a member of the Penn Vasculitis Center. She received the Vasculitis Foundation's Fellowship Award to support her dedicated training in clinical management and research of vasculitis, and since then has been the recipient of several research awards, all focused on vasculitis. So welcome, Dr. Ree. We're so happy to have you with us today.
Great, thank you so much. I'm so excited to be here to talk about the microbiome. Let me just share my screen.
And while you're doing that, if you would also, I have to ask you if you would let us know if you have any disclosures.
Yes, I'll definitely include that. But
Okay.
All right, great. Well, we'll go ahead and get started. I'm Rennie Ree, excited to talk about the microbiome, your hidden helpers. Thank you to the Vasculitis Foundation for inviting me and just their general relentless dedication to educating and supporting the broad community of patients with vasculitis. So really terrific to be a part of this. And my disclosures are here related to this topic. I just have some research grants from the National Institutes of Health and Rheumatology Research Foundation. So I will be discussing the microbiome and human health and disease. I will then talk about some research that I've been doing in ANCA-associated vasculitis, and particularly the nasal microbiome, and then end with a few misconceptions and practical takeaways. My main goal is to really help you appreciate the microbiome and its role and why I think it should be studied in vasculitis. But we're still in the early stages of understanding the microbiome. So I will caution that at this time, there's really no evidence to make recommendations for changing your microbiome to treat vasculitis. And in fact, in other conditions, we've seen at times that there can be more harm than good when we try to change the microbiome. And I'll give some examples of that at the end. But I think regardless, I think we are on the verge of understanding a whole new aspect of human health that I think is really exciting. And hopefully you will think so too. So we're going to start with what is the microbiome and why is it so important to our health? So a biome is a distinct community of living organisms. And if you were to take planet Earth, from far away, you will see a beautiful sphere of blue and green. And only when you zoom in will you suddenly see that it is covered with complex living organisms, which are intricately intertwined with each other and its environment. So similarly, if you are to zoom in on the human body with a microscope, you will actually find that there are trillions of microorganisms such as bacteria, viruses, fungi that live in and on our bodies. And what we're understanding now is that these microorganisms don't just reside on us, but they interact with us and are very important to our daily function. Here are some facts about the microbiome. I think they're fun. Maybe you might find them gross, but regardless, I think they're very interesting. So a few things. If you were to count the number of microbial cells on the human body, there would be an equal number of microbial cells to human cells. If you were to take all of those cells, put them together and weigh them, they would be about 2.5 pounds or have a total volume of 3 pints. Now, of course, even though cells are the same number, the actual mass is different because microbial cells are much smaller than human cells, but it's still an astounding figure. If we're looking at genes, for every human gene, there are up to 300 non-human genes that are on our body. And then across individuals, there's quite a bit of variability in the microbiome. So while the human genome is 99.9% the same across all people, the human microbiome is quite different and only about 10 to 20% similar between individuals. And some have wondered whether that difference in the microbiome could in some ways contribute to why certain people are prone to certain conditions and diseases. The microbiome rapidly develops in the first few years of life after birth and then tends to stabilize over time, but the microbiome can change in response to things like diet, antibiotics, and the environment. When we think about the microbiome, we tend to think about the gut, and this is indeed the largest reservoir of microbes on our human body. And there's been a lot of research really centered around the gut microbiome, which has shown that actually there's so many important functions that the bacteria have that enable us to live day to day. For example, the bacteria can help us break down food and its components. For example, human cells only have about 20 enzymes to break down carbohydrates. But one type of bacteria in the gut has over 260 carbohydrate digesting enzymes. So those bacteria give us capabilities that we otherwise don't have. The bacteria also produce molecules like short-chain fatty acids, which help support the health of our gut cells. They can protect our gut from harmful or pathogenic bacteria, and they can synthesize vitamins like vitamin K. And then we'll talk a little bit about how important the bacteria are in shaping and regulating our immune system. But beyond the gut, there are microbes throughout our body. Pretty much any surface that is in contact with the air has microbes in it, and from the airways, the mouth, the skin, and many other areas. And the communities of microbes in all of these different locations are very different, just like on Earth, kind of the organisms in the ocean versus in the mountains, the desert, are all different and have been adapted to those specific environments. Likewise, the microbes in these different environments on the human body can differ. So if the microbiome is essential for human health, then it begs the question whether alterations in the microbiome can contribute to disease. And many investigators over the past 15 years or so have been very interested in studying about the microbiome in a variety of diseases. And they have found connections between the composition of the microbiome and disorders such as obesity, cardiovascular disease, chronic inflammatory diseases, cancer, stress, and even neurodegenerative disorders. But most of these studies have really only found links, but have not actually proven causation. They have not actually demonstrated that this microbiome is causing disease. And so I think this is still kind of where the field really needs to move beyond links to actually finding causation to demonstrate that this could potentially be a target for treatment. And just a reminder, why correlation is not causation. So I think an example is if we were to make the correct observation that the more the consumption of ice cream occurs, that there is also more sunburns occurring. That would be a correlation, but of course we know that the ice cream is not causing sunburns. In fact, there's another factor that's associated with both of these, the temperature, that is resulting in this relationship. And so similarly, we may see changes in the microbiome in people with diseases, but it doesn't necessarily mean the microbiome is causing the disease. There could be some other factor, for example, maybe changes in the immune system that could be contributing to both. The other thing with doing research studies in microbiome is trying to understand the direction of causation. So for example, if we were to see fire trucks, we would also notice there are more fires. And the more fire trucks there are, the bigger the fires. But we know, in fact, it's not the fire trucks that are causing the fires. It is the fire that is attracting, signaling the fire trucks. And so similarly, the microbiome may be causing disease or it may not be causing disease. Maybe the disease is causing the changes in the microbiome. So I think that's where we need to do better studies of looking for cause and effect. And the best ways to do this are either through studies in animals, usually mice, or in clinical trials in humans. And so I think one example of really convincing evidence to demonstrate causality was shown in a study of mice that really ultimately helped researchers understand the importance of the microbiome and our immune system. And there's been quite a few studies. I'm just giving this one example, but there's been many, both in people and in animals, that have demonstrated this relationship. But what researchers found, I think the really quintessential study that was shown was that when they took mice that were germ-free, they were raised in a sterile environment, had no microbes on them, their immune systems were underdeveloped. And when they took those same mice and colonized them with microbes, like re-added back and reintroduced the microbiome, those deficits in their immune system reversed. Another interesting study has come out in terms of metabolic diseases. So similarly, they did studies in germ-free mice, and they did something called fecal transplantation. And that is basically taking stool, poop from another mouse and giving it to the germ-free mouse. And when they took fecal matter from an obese mouse and gave it to the germ-free mouse, that germ-free mouse became obese. And similarly, when they gave the fecal transplant from a lean mouse to the germ-free mouse, that mouse then became lean, which is just like really kind of fascinating to think about that huge of an impact that the microbiome would have on certain outcomes. And there's been a lot of links in humans too, just looking at things like early antibiotic use in childhood and development of obesity and other metabolic disorders later on in life. Now, in terms of studies in human, right, we ultimately want to, we're not mice. We want to be able to demonstrate that certain things are happening in humans too. I think the one example that has been shown time and time again is in a diarrheal infection known as Clostridioides difficile infection, or C. diff. This is a bacterial infection. It tends to occur after antibiotics when the antibiotic has disrupted the normal gut bacteria, and individuals who are unable to restore their indigenous microbiota, they may be more prone to this C. diff infection. And many times we will treat with antibiotics, but patients will have recurrent infections. It becomes very, very difficult and challenging to treat. And so studies have been done of fecal transplantation in individuals who have this recurrent infection. And it was shown that when you take these patients and you give them a fecal transplant from a healthy donor, that they were able to restore their microbiome and completely get rid of the infection, which is just really amazing. I mean, it's actually a little crazy when I initially heard about it. Who knew that every day you're flushing down the toilet to cure to someone else's disease? But in fact, this was shown to be beneficial and is now an FDA-approved treatment for this severe infection. The other thing that this taught us is that it's not just so much about getting rid of certain bad bugs, but it's also about restoring the healthy community. And it's really that whole community that is really important to human health. The other thing is that in terms of the gut microbiome, what we eat is important too. So they say you are what you eat, or maybe it's what your microbiome eats. That's really important. And the types of food that we eat can increase certain types of microbes. So some studies have shown that perhaps a Western diet can decrease healthy microbes, and that may be associated with a higher chance of inflammation. On the other hand, Mediterranean diet may lead to an increase in healthy microbes, and that may overall provide some health benefits. Another example is just human breast milk. There's actually many compounds in human breast milk that are undigestible to humans, but do allow certain bacteria to grow in the infant gut, just demonstrating really the importance of having a healthy microbiome. So we know that the microbes are essential to the healthy development of the immune system and constantly interacting with our immune system, which led researchers to study the microbiome in autoimmune diseases. And in autoimmunity, although for a specific individual, we often don't know what causes it, what many believe happens is that there is a complex interplay of alterations in the immune system, genetics, and the environment and it could be hundreds of different factors that arise, ultimately lead to these very rare diseases. And so many researchers have been studying the potential impact of the microbiome in autoimmunity. And there's been many autoimmune diseases in which this has been studied. Inflammatory bowel disease is perhaps the most well-studied. And in that, researchers have found that really the microbiome may be playing a role, but also the inflammation may also be altering the microbiome. And it could be this two-way street, this bidirectional kind of relationship that is ultimately contributing or even propagating disease. So in summary, the microbiome is a complex community of living organisms on the human body. It's essential to human health, including the immune system, and it's been linked to many diseases, although scientists are still trying to figure out if the microbiome is causing disease. And next, I wanted to talk about a research interest of mine in terms of the microbiome and in ANCA-associated vasculitis. And the idea of microbes and vasculitis is certainly not new. So there are several types of vasculitis that have been linked to certain microbes. These are only in a subset of patients. So I will say if you have one of these types of vasculitis, it's most likely you will not have any of these infections. But in a subset, we have seen that these infections can cause these types of vasculitis, and getting rid of the infection helps get rid of the vasculitis. So hepatitis C virus and cryoglobulinemic vasculitis, hepatitis B virus, kind of prior to the hepatitis B vaccine, used to be an important cause of polyarteritis nodosa, much less common nowadays. And in IgA vasculitis, streptococcus infections. And for many years, people have been interested in nasal bacteria and ANCA-associated vasculitis. And the interest in the nasal cavity comes out of that clinical observation that there's a lot of inflammation in the nose and sinuses that happen in patients with ANCA-associated vasculitis. So many of you are probably familiar with ANCA-associated vasculitis. It's a small vessel vasculitis, and it can often affect sinuses and nose and many other organs as well. Sino-nasal inflammation can occur in up to 90% of patients. It's often the first manifestation of disease, and it is associated with a higher risk of future relapse. And among the three types of ANCA-associated vasculitis, one of them, granulomatosis with polyangiitis, or GPA, is the one that is particularly associated with very severe and destructive sinus and nasal disease. And so a lot of the early studies were really focused on that population of patients with GPA. And they, initial studies in the 1990s found some relationships, some links with a bacteria that's called Staphylococcus aureus. This is the bacteria that's kind of ubiquitously found in everyone. It's often seen in the nose as well as in the skin. And what they found was that they could see the presence of this bacteria in the nose more commonly in patients with GPA than in healthy patients. And among those with GPA, they found that those who were colonized with this bacteria in the nose were more prone to developing relapse. Again, this is correlation, but we don't know yet if that bacteria is actually causing disease. So that led the researchers to do a clinical trial of an antibiotic, which was associated with a reduction in relapse. Despite this study, however, antibiotics continue to not be a main treatment for GPA, and there's a couple of reasons why. In that trial, there was a high rate of dropout just from side effects of the antibiotic. There are a lot of concerns, long-term safety concerns about chronic antibiotic use and the development of drug-resistant bacteria. And there were some questions about the relapses that happened in that study. Many of the relapses were limited to the ear, nose, and throat area, and some wondered, were they truly relapsed or were they just preventing infections? And then lastly, the antibiotics that was used is also known to have some, or is thought to have some anti-inflammatory effects. So they wondered perhaps it's an anti-inflammatory benefits that led to this, this finding. So, in that context, I got very interested in studying the nasal microbiome and ANCA-associated vasculitis. And there were still a lot of questions that were unanswered. And we talked about the community. And beyond that one bacteria, what changes were occurring in the broad community of microbes? And why, in general, are patients with GPA so susceptible to inflammation in the nose and sinuses? So we wanted to leverage some existing technology that had come out through all of those studies of the microbiome, including these kind of advanced sequencing technologies that were becoming more and more accessible to people and doing research. And to do this, we many years ago got a grant from the NIH to study sinonasal disease and ANCA-associated vasculitis. So we, at every visit, collected nasal swabs, patient-reported outcomes, and other clinical data from patients who were coming to the Penn Vasculitis Center who had a diagnosis of GPA. And eventually, we also started recruiting patients with eGPA and MPA, and recently relapsing polychondritis. And it's really just through the tremendous efforts of all of the rheumatologists at our vasculitis center, Dr. Merkel, Banerjee, Shrai, and Naomi Amudala, and many of our research staff, that we were able to recruit quite a large number of patients, over 300 patients with multiple visits, and then be able to use these samples to really further examine some of these questions. So some of the things that we wanted to look at is, well, what about the bacteria in the nose? Not just Staph aureus, but everything else. Are they different from patients who don't have GPA? And so we did an initial approach, just looking at a group of patients with GPA, comparing them to healthy patients who don't have GPA. And what we see here is each of these colors represent different types of bacteria. And you can see that patients with GPA had many different, more different types of bacteria than patients who didn't have this condition. And then the other interesting thing we found was that people with GPA who were on treatment were actually more similar to healthy patients, whereas those who were off treatment were different. And so, kind of raised a lot of interesting questions. Could it be that perhaps the treatment, there was still some subclinical or mild inflammation that was occurring in those off treatment? Was there something about the actual medication itself, or potentially some other factor that was associated with it? It answered some questions and raised new ones, which led us to then study this further. Instead of just looking at a single time point, which is what the previous study did, we wanted to look at multiple time points within an individual. And by doing this more of a longitudinal study, we can look not just at what happens at the time of relapse, but we can also look at what happens prior to relapse, that pre-relapse visit, months before people develop symptoms of their disease. And this also enables us to kind of control for the heterogeneity across different people, because our microbiome is so different across different individuals. But by kind of using that same individual and looking at changes over time, we can kind of use a person as their own control, in a way. And so we asked, do the bacteria change prior to and during relapse? And what we found is, yes, that there were some changes happening prior to the onset of relapse. So here you see this graph, and it shows these 5 different types of bacteria and their relative abundance, the proportion of each of these bacteria in these groups. And if you look at the people who were in remission, the people who had quiet disease over many time points, and you compared them to the time point of relapse, you actually wouldn't have seen a difference. They would have looked pretty much the same. But because we're able to do this longitudinal study, we were also able to capture that pre-relapse visit. And it was that pre-relapse visit, it's about 3 to 6 months before they flared, where we saw big alterations in some of the bacteria, particularly in the Corynebacteria and the Staphylococcus. So we got very interested in this relationship. And in addition, this new bacteria, which was a bit of an unexpected finding, we kind of explored that a little further. This is not a bacteria that's been well studied in vasculitis and in general. And I've subsequently learned it's actually very difficult to grow from a swab. But what we found is when we looked at these bacteria, they were increasing leading up to a relapse. And we were also seeing that they were associated with ANCA titers, which is just a blood test that that has been associated with the disease. So some kind of initial intriguing findings about this bacteria, but I think, again, the caution being, we still don't know if these bacteria are causing or just responding to inflammation. We will return to this as we are now trying to do other studies to get a little closer look more at causation rather than correlation. But what about the human side of things, right? We're focused on the upper airway. We're looking at all the bacteria there. But what about the human cells? What about those nasal cells? And could there be changes in the nasal cells that are altering the microbiome? Maybe it's not the bacteria. Maybe it's our response to bacteria that is altered. And I mentioned before that inflammatory bowel disease in the gut, they have seen that really there's this bidirectional relationship between the microbiome and gut inflammation. So while we're doing this, we decided we also want to study the nasal epithelium, that layer of cells that line your nose and your sinuses. And it is really quite interesting. I've learned a lot about the epithelium kind of in this process, but there is really this, the epithelium is this lining, as I mentioned, and it's not just a physical barrier from our environment to our immune system, but it is very immunologically active. It is doing a lot of work to maintain a healthy balance. And it's constantly interacting with our immune cells and with the microbes and various other things in our environment. And, all that snot that you blow out every day is actually a very complex coordinated event of many different cells and compounds and activities to help constantly replenish and refresh the epithelial lining, and even actually that the mucus in our nose has important nutrients that help certain bacteria to thrive. So we wanted to study this layer from our patients, and we did this by taking those same nasal brushes, or a different set of nasal brushes from patients, again, focusing on GPA just because they have the really severe sinus and nasal inflammation, and looking at people who relapsed versus those who didn't to healthy controls. And we submitted those samples to a lab where they are able to do all of this fancy kind of molecular sequencing to look at markers in those nasal brushes. And what we found again was that there were changes that were observed that were detectable many months before the onset of relapse. And this is just a heat map of multiple different genes. Each row is a different marker, and each column is a different time point. And when we took a group, we looked at the relapsing versus the non-relapsing patients, we found that there were certain markers that were increased, right? The darker boxes mean there's an increased level of that marker. in the patients who relapsed compared to those who didn't. And a lot of these markers were present even at that pre-relapse event, many months before the onset of relapse. So kind of really, really interesting findings, a small number of patients, so hard to make any kind of conclusive comments about this, but really kind of was like a carrot stick for us to kind of keep pursuing this. And perhaps there's something really important going on in that upper airway that could give us clues into why people are relapsing with GPA. To understand this more, I mentioned how, a lot of these are correlation and not causation. And so now what we've been doing is we've been working with an ear, nose, and throat scientist who has developed a method to take cells from a patient swab and then grow them up in the lab. And there we can kind of remove them from their environment and study them in isolation, and then be able to do all sorts of experiments with them, including perhaps, putting bacteria on those cells and seeing what happens. So we have been collecting these nasal brushes from patients with GPA and eGPA and have been growing them up and studying them. And it's been really interesting, and I won't go through all this data, but what we've been finding in general with our preliminary data is that so far there are a lot of differences between patients with ANCA-associated vasculitis from people who don't have the condition. Differences in the types of cells that are present, in the function they have, and the proteins they produce. Even just, without putting any bacteria on them, just looking at them by themselves, they are different. Here you see, these are like pretty pictures of different of the cellular composition of the cells, and each color represents different cell types. And you can kind of see that the patients with GPA and eGPA were different from the control patients. So the other thing we've been doing is trying to culture bacteria straight from the nasal swabs and be able to study those in greater detail as well. That has been a very lengthy process, actually. Actually took us a few years to get this right because it was so hard to grow up that bacteria that I mentioned, the Corynebacteria, and it took a lot of different iterations to get this done. But we have been able to develop a process where we take a swab from a patient and then grow up the bacteria in the lab and isolate them. And by isolating those individual types of bacteria, we can study each of them in greater depth, including sequencing their DNA to understand what type of bacteria are present and what can they do, as well as to be able to do experiments in terms of seeing how they interact with each other, and also how they interact with those nasal cells that we've isolated as well. So a lot of ongoing work in this area, which is it's really been exciting and a fun thing to work with others on as we are trying to learn more about this. And I think for the interest of time, I won't go too much into this, but it's kind of this idea, again, of taking a swab, growing up different bacteria, and sequencing those bacteria to study them further. And we can, through the help of bioinformatics specialists, we can kind of analyze the different bacteria and their different functions and see if there's any similarities across certain people who have certain outcomes, like relapse versus no relapse. And then there's infectious disease specialists I've been working with as well, who are helping to do studies, kind of culturing bacteria together and looking at how they behave when they're in the presence of other types of bacteria. So really kind of still in the early stages of a lot of this, but I think for me has been really exciting to start exploring really kind of an unexplored area in ANCA-associated vasculitis, trying to understand the intricate interactions between microbes, sinonasal cells, and the immune system, and see if this can eventually lead to perhaps a better, more accurate, reliable marker that can help us know who's prone to relapsing and who's not. and perhaps down the road in the future to see if we can actually target some of these interactions as a therapy. The other area of research related to this has also been in just understanding the patient perspective when it comes to sinus and nasal disease and ANCA-associated vasculitis. And this is a study that we recently launched through the VPPRN, the Vasculitis Patient Powered Research Network. So if any of you have a diagnosis of EGPA, GPA, or MPA, we would love for you to be involved in this study. We are trying to know, what does the nose know? And this is an NIH-funded research study, and we're looking at how symptoms in the nose and sinuses impact patients living with ANCA-associated vasculitis and how these symptoms are related to relapse or flare of their disease. So if you are interested, you can go to this QR code. This can be done by anyone living in any part of the world. All it does is require you to complete some questionnaires every 3 months. And through that, we'll be able to kind of look at a lot of these questions we have. And whether or not you have sinus or nasal disease, we would love for you to be involved, because actually studying the people who don't have sinus disease or nasal disease would be interesting as well. And this has also been a wonderful experience working with some patient research partners. Mary Gibson and Darren White have just been phenomenal research partners in this, have been involved from the very beginning, kind of designing this study, helping to really provide the patient perspective about the sinus and nasal symptoms. They even helped generate this fun study name, V-SNIFF, and as well as this logo. So shout out to them for their involvement in this. And if you have any questions about the V-SNIFF study, contact Christine Young, and here's her information as well. So I'm going to end with a few misconceptions about the microbiome and some practical takeaways, and then I'll be happy to take some questions. I would say if there's any slide that's the most important slide of this talk, I think it would be this one, actually. And it is about some misconceptions about the microbiome. I do think the microbiome is really fascinating and exciting, but we just have to be careful about how we interpret this. First misconception is that all bacteria are harmful, and hopefully I've convinced you the opposite. Actually, there are many bacteria that are really essential for our function. Another misconception, there is one perfect microbiome, and in fact, we actually see that there is not, and what is healthy really can vary across individuals. We've also talked about how the microbiome does not only exist in the gut, it exists throughout our different surfaces of our body. And there are researchers who are focused on the skin microbiome and the lung microbiome, and et cetera. Another misconception, antibiotics only kill bad bacteria. Actually, unfortunately, antibiotics kill beneficial bacteria as well. So we just have to be careful in the use of antibiotics. They are, of course, important treatments in the right situations, though. Another misconception is that supplements can quickly fix your microbiome. I wish that was the case, but unfortunately, restoring the microbiome is quite complex and depends on multiple factors. Probiotics always improve health. Actually, the effects of probiotics can differ across individuals and is not a cure for restoring balance. I think this is one area that I just wanted to talk a little bit more about in terms of probiotics. So probiotics are cultured strains of bacteria that are often in a pill form that we can take. And there have been some studies showing some benefits, for example, in help reducing antibiotic-associated diarrhea. But there have also been studies demonstrating a detrimental effect of probiotics. In this study, I just wanted to highlight this one. This was really a well-done study in which they gave antibiotics to some people as well as to mice, and then deeply evaluated the gut microbiome. They then took these people who got antibiotics and then randomized them to either get probiotics, autologous fecal transplant, so they took their own stool before antibiotics and gave it back to them, or just spontaneous recovery, meaning they didn't give them anything. And actually, the group that got probiotics had the worst outcome. Those people had a marked delay in recovery of their gut microbiome after the antibiotics. And the group that did the best was the autologous fecal transplant, people who got their own healthy microbial community back. So, once, studies like this make us worried about potentially the damaging effects of probiotics. And really, many health organizations have put out statements just cautioning the safety of probiotics, which really have not been adequately assessed. There have been case reports too of bloodstream infections occurring from strains that are found in probiotics. And in fact, sometimes we really try to caution this in immunocompromised patients. And there have been several studies, including another clinical trial of probiotics in severe pancreatitis, which found that it was associated with worse outcomes, including more mortality. So, so just a word of caution, not something I would use, not at all something I would use to treat vasculitis at this time point. What can we do with all this information? I mean, I think I hopefully gives you more motivation to be eating a well-rounded diet and making sure it's diverse and fiber rich. We talked about the Mediterranean diet and how there's been a lot of cardiovascular benefits that are associated with that. Should be more cautious in our use of antibiotics. Absolutely, you got a bacterial infection, you absolutely need to take the antibiotic. It's life-saving, but also I think, it's probably the fault of our healthcare professionals is that we often can be very quick to prescribe antibiotics. And so there's been a lot of trying to pull back and rein that in a bit more because we are seeing some detrimental effects of just overuse of antibiotics. Please continue the treatments that are recommended by your healthcare professionals and support research. And with that, I will conclude. I just want to say it really, some of the research that I presented really took a village to make this happen. And I am by no means an expert in microbiome on our campus. I had to reach out to other people who are experts in the field and experts in bacteria and genomics and lots of other different areas to bring them into the field of vasculitis and get them interested in these questions. So really thankful to be in this environment where there's so much collaboration going on. Also to our wonderful patient participants, some who are, I think, on this call who have been a part of this research and have just been wonderful to work with. We really appreciate all of you. And also to all of our funding through the NIH and the Rheumatology Research Foundation. So big plug for research. You guys are all beneficiaries of research. I think today our ability to manage vasculitis is leaps and bounds from where we were 20 years ago or even 10 years ago. And that's all due to the patients who volunteered, to agencies like the Vasculitis Foundation, the NIH Rheumatology Research Foundation, who've supported this work. And so with that, I just want to thank you. And one more plug for the V-SNIFF study if you are eligible and interested in participating. And that's it.
Okay, thank you so much. I feel like I learned so much about microbiome. I wondered because I didn't know very much about it, just what people were saying. If you would mind, you could stop sharing your screen so we can move on to some questions, Dr. E. We appreciate your really great presentation that you did for us today. We've not gotten very many questions, but we do have a few. So I wanted to start with the first one, which is, one patient wondered, why is your focus on ANCA associated and not on other non-ANCA types?
Well, I think it's just a matter of time and resources, but we do believe that these studies will be different based on the underlying disease. So we wanted to try to study a similar group of patients. And just because the nasal microbiome really at the time of when we first started this many years ago really hadn't been well studied, just seemed like a really ripe opportunity to go into that. So I have done a study of the stool microbiome in large vessel vasculitis, in giant cell arteritis and Takayasu's arteritis. And so that is still, I haven't gone into that really just because we don't have any data yet, but we are hoping to analyze that data in the near future.
Okay, great. Well, that sort of explains it to everybody, and I'm happy to hear that you're also studying the other forms. One of the questions that came up today is, do you think that, I mean, and you alluded to it in your presentation, but I guess the specific question is, how can understanding the nasal microbiome, I mean, uh, could it possibly indicate where relapse might be coming? I think you did answer that, but I just wanted to just reiterate because everybody wants to know how we can tell if we're about to flare or relapse.
Yeah, honestly, I find patients know before I do, and what I had heard from patients, and I know many other vasculitis specialists have heard as well, is there's often this common story where they start developing symptoms in their nose or their sinuses, and then months go on, and we're not quite sure what that means, but we continue to follow, and then it starts propagating into other areas, their joints or their lungs or perhaps their kidneys. And we've seen that kind of trend over time, which I think is really interesting. Like why does it tend to start in the nose? Even if it doesn't start in the nose, eventually ends up there and eventually involves the nose and the sinuses, whereas many other types of vasculitis don't. I feel like there is a signal of something important that can help us understand relapses in the nose and sinuses, which is why I'm really interested in studying and understanding that better.
Okay, thank you. And one of the questions was, is anything known in reference to using probiotics in vasculitis, pro or con? And you answered that in detail, so I'm not going to answer you to put that out there again. You did great information for all of us. And I honestly think, I mean, I personally did not know that, and I have taken probiotics, so it's good to know that there are definitely cons to using probiotics as well. One of the patients asked, most of the discussion is related to GPA. What about MPA? I've had 2 relapses or flares elevated, and says a lot of the ANCA titers, but both followed prolonged and severe sinus inflammation.
Hmm. Yes, we have been studying, we are definitely interested in studying MPA, all of them really. And I've heard that too. I feel like we tend to think of people with MPA as not having sinus or nasal inflammation, but I've seen that, like people we kind of labeled as MPA, and then actually, over time we are identifying more of their sinus and nasal disease. And I wonder if, in any case, I totally agree. MPA is being studied as well. We just, in terms of numbers, we see a lot more GPA, so it was just a lot easier to start with that. But we think it's definitely worthwhile studying all the different types of ink-associated vasculitis.
Okay, thanks. I was interested too as an MPA patient. I wondered about that. The next question is, are you aware of any connection between GCA and the nasal microbiome?
I am not aware of anything related with giant cell arteritis. I didn't include this in this talk. There were studies in the past, I believe it was out of Cleveland Clinic, where they were actually trying to study the temporal artery microbiome. The temporal artery is an artery on the scalp that we often biopsy to diagnose giant cell arteritis. And there were investigators who were using these sequencing technologies to pick up on hints of bacteria there. I don't think any more has come out of that, but I think it was just interesting to see that potentially there could be, different microbes present there. But I think really an understudied area at this point.
Okay, that's interesting. Maybe we'll hear about that in the future. I was going to ask you, what do you think is the biggest surprise or something that you've learned about your study in the microbiome?
Um, I guess, I mean, we started off really interested in Staph aureus, and I still think that Staph aureus may be important, but what we did by kind of opening up the possibility and looking at the whole community of bacteria and other types of bacteria, just finding that Corynebacteria was really interesting. And I've just it's a really hard bacteria to grow, which is why I think people don't really study it very much. And I just wonder if there's something important about that bacteria that is underappreciated right now. But I feel like every time we do these things, we think one thing and then we find something else, and this takes us down a different kind of pathway, which is really fun, actually. I mean, it makes it interesting. We follow the data and then we just keep pursuing it.
It does sound interesting. One of our questions that came up today, and this is probably something a lot of us have thought about doing, but or do it, but would using a neti pot and doing daily sinus rinses help our microbiome, or is that not good? Do you know?
Um, I guess I don't know the answer in terms of its effects on the microbiome. I will say I generally recommend using it in people who have constant, like, chronic sinus congestion, crusting in their sinuses and nose, just from a symptomatic relief standpoint. But I do think that there is, just simply from all the inflammation that causes damage to that tissue lining, sometimes the ability to kind of clear away all the debris and the bad bacteria can get compromised. So using things like those sinus rinses or neti pots, those irrigation systems, may help just supplement what normally our nasal cavity should be able to do, which is just like sweep things away and get rid of harmful things. So I feel like I generally recommend it anyway, just because I do notice a lot of patients benefit from it.
Still a good thing to do, so thanks. Somebody else asked, how do we know, or do you know how medications that are taken for the various types of vasculitis, how they affect the microbiome?
That's a good question. There have been studies actually in rheumatoid arthritis, looking at methotrexate. We use methotrexate in some types of our vasculitis, many of our different types of vasculitis. At some point, we've used methotrexate. Rheumatologists love methotrexate.
Yeah.
And it's really commonly used in rheumatoid arthritis. And they have seen changes just simply from methotrexate that happen in the gut. So that's another whole area of the microbiome, in terms of just medication effects there. So I think there is probably something there.
Well, it is really interesting to learn from you today that there are so many other places that the microbiome exists, because we kind of only hear about the gut from the rest of the world. But I did want to give you a chance to talk a little bit more about research and how it could potentially help patients with vasculitis. Is it tied to understanding relapse or other ways that it could help patients in a direct way? I mean, we need to know. I think it's important for us to know why we should participate in research.
Yeah, no, I appreciate that question. And um, yeah, I will say, as somebody in vasculitis, studying rare diseases is super challenging. Actually, I will say, in the early course of my training, I was advised not to study vasculitis because I was told it would be too hard. I'm glad I didn't listen to that person, but I think that, or just being able to stay afloat in the world of research is challenging enough, and it gets more challenging when it comes to rare diseases. But I think that what has made it really fun for me is being with patients who also value and are excited about research and are just so generous with kind of their time and willingness to be a part of it, knowing that it doesn't directly impact you today, right? It's not going to benefit them directly, but it is hopefully going to have a positive impact on the future in terms of future patients who develop this disease, just as I think everyone today has benefited from people in the past. So it's not for everyone, but I think that if you are in a position or, are at a place that has a vasculitis center, oftentimes you need to be at these major centers because when it comes to rare diseases, you need numbers. And so only places that see a lot of that disease are able to really study it in depth. So I've been lucky enough to be at a place that has many people who are studying vasculitis or seeing patients with vasculitis. And so we all work really well together. It's a great kind of environment in terms of other clinicians and just other scientists who are like excited about research. And many of the people I collaborate with, like other scientists, have never even heard of vasculitis. So they never would have even wanted to study vasculitis, but because we are there, we kind of introduce them to the field and they get excited about it too. So I think that, I'm a big proponent for it if that's something that you're interested in. Even just donating money, even, your time volunteering for those who are able to financially support research as well. I think I really encourage every little bit counts.
Well, thank you for being our vasculitis research champion. We're very excited to have you working on this project, and specifically because we're doing this recording in March, and March is Autoimmune Awareness Month. Is microbiome research being done in many other autoimmune diseases, or I know you touched on that a little bit. I just thought I'd give you a chance to reiterate.
Yeah, there definitely are other many diseases where people have studied the microbiome. So I feel like we are just skimming the surface right now, but it's really exciting. And I anticipate there's going to be a lot more that we're going to learn the next few years.
Okay, great, thanks. And I just, before we finish up, I did want to say that if in case you want to address the SNIFF study a little bit more. I saw the QR code, which when the recording is up, people can go back and find that. It's also been posted, the link in the chat, if anybody wants to look in there. But is there anything else you want to say about the SNIFF study to get people to do it? Like, are people reluctant to participate in this sort of thing? Is there a lot of time involved?
I mean, the questionnaire should take about less than 10 minutes. It does require that you complete them every 3 months for 2 years. So we're really asking, like, if you sign up to stay in it and do it every 3 months, because we are trying to understand those changes over time and just relationships with vasculitis. So I know it's tough. I know everyone's busy, but the more people that we get to participate, then the better chance that we'll be able to find something meaningful.
Well, great. So we greatly appreciate the time that you spent with us. Because you spent so much time talking, which was wonderful, about VPPRN, I just want to share a little information for everybody to make sure everybody has access to it. So they should be able to see my screen now. And I just want you to be aware that VPPRN is part of the Vasculitis Foundation. It's the Vasculitis Patient Powered Research Network. It's not a lot of work to be registered with the VPPRN. They, like, twice a year, they ask you to complete biannual forms. I think they take maybe 10 minutes. I'm one of their patient champions. I would just encourage you, if you're not associated or have not already started with VPPRN, to just go to their website. There's a dashboard on there that tells you so much about all of us and the different types of diseases and the research that's being done. And it's a patient-powered, driven research. And so there's basically no other way for them to get the information than from us. So I'd like to ask you all to do that. And then if there's anything else that we would want to say today, it's just simply that thank you so much for your time, and thank you to the Vasculitis Foundation for supporting these educational webinars.
Thank you so much.
All right, thank you. Goodbye, everybody.